The Conversational Flow, a KCAS podcast.
Hello, and welcome to the 7th episode of The Conversational Flow, an official podcast of the KCAS Bioanalytical and Biomarker Services. KCAS is a bioanalytical CRO serving the pharmaceutical and biopharmaceutical industries for over 40 years. Unlike KCAS' other podcasts, the Weekly Bioanalysis, which features news and topics more generally associated with the world of bioanalysis, the Conversational Flow focuses more on the niche of flow cytometry and its role within the greater industry. My name is Adam Cotty, and I've been in the flow cytometry space for over 15 years. I'm here with my co-host, Brian.
Thanks, Adam. My name is Brian Weil, and I've been in the flow cytometry space for over 10 years at this point.
Brian and I are members of the growing leadership and scientific advisory team at KCAS. Either or both of us are available to answer any questions you may have regarding this podcast or any of KCAS services. We're also joined by a special guest today, David Ambrose, an associate director at KCAS Bio, who will join us in discussing receptor occupancy assays. David, why don't you introduce yourself?
Thanks, Adam. Hi, everyone. My name is David Ambrose. I'm an associate director of flow cytometry services here on the operations team at the Philly site for KCAS. I've been with the team over 2 years now and been working in flow cytometry for over 17 years. And it's great to be here today with you guys.
Thanks, David. Looking forward to our discussion. While our KCAS headquarters are located in Kansas, with additional location in Europe, we are both here in Philadelphia, actually all three of us, and Jeremy, our producer, is in Missouri. So we truly are all over the map. We are elated to have you listening to the 7th episode of our podcast, which will be available virtually everywhere. Wherever you choose to find and play our podcast, you will likely find the Conversational Flow. We welcome all of you, whether you're joining us for the first time or if you're a regular listener to any of our other podcasts. Today's episode will be a review of the latest news and resources and then a focus on a topic of our choosing before discussing any feedback we receive from you. We're constantly looking for topics and we'd be happy to discuss something that you want us to cover. So again, we're exhilarated to have you here and looking forward to a fun episode today. To kick us off for episode number 7, Brian is going to go over news and resources before diving into today's topic. Brian?
All right. Thanks, Adam. In terms of medical research news for this month's episode, we picked out 2 stories that aren't as medical as we would usually do. But the first one is on Roche's R&D rethink. And we've seen a lot of this from Big Pharma recently. So that's one of the reasons that we picked it out. But the reason I picked out this story is that they are looking at their overall pipeline of drugs in development, and then illuminating those that are either no longer commercially, reasonable from what they are projecting, or really shifting around which ones are going to be sort of best in class and most relevant.
Right. Looking to refocus high probability of success and high impact. Seems in part driven, some of the stuff I was reading in the loss of COVID sales. And so they had done a lot on testing, things of that nature. And so, as you might imagine, the demand for that sort of thing is reducing. And one of the things I found interesting in researching a little bit of the Q1 of Roche is didn't really think about this previously. Their sales improved by 2% over quarter 1. However, it also depends on how you look at it. That's improved in 2% when you account for currency fluctuations, whereas, if you're not accounting for that, their sales went down by 6%. So just something international learning for myself there. They have started this culling of programs. The 20% wasn't all just this quarter. It started quarter 3. And they had recently acquired, Carmot, for the weight loss drug. So certainly big changes happening here for Roche.
And the piece that always sort of concerns me or makes me really think about what this means for patients overall is, as they're culling these molecules that may not be as commercially viable, are rare diseases or orphan diseases the ones that potentially fall into that bucket, and how will this impact sort of new drug development? So are there now targets that are sort of no longer considered a good path because of this culling process?
Now, the ones that were impacted most recently, they had an oral RAF kinase inhibitor. So it was a phase 1B for cancer. They also had a bispecific for colorectal cancer. So looking at a tumor-specific antigen and CD3 are the arms for that. And then they also had a early-stage psychiatric drug that they were cutting. It does seem like the cancer ones, at least the indications don't seem to be particularly rare. However, they did mention that will continue treatment at least on some, if not all of these, despite the fact that they're discontinuing. So for those clinical trials, they maintain with the patients there. So that's definitely something good to see.
Sure. And, as with all drug development, there's definitely a game of chutes and ladders where a molecule will look good for a while and then potentially look bad. But then, as we've seen with some of the Vants and other companies, sometimes they get a second crack at making it through the process and getting through to patients. So,
it's amazing, right? Yeah. They always seem to come around, these targets. I remember prior being at Janssen and working on a molecule when I first started, and then 10 years later, all of a sudden it comes back around. So, yeah, I certainly understand what you're saying there.
That's it. And speaking of coming back around, that's a good segue into the second paper that we looked at, and that was around BMS's deal with Solaris. So I want to pick that up because Solaris is right in our backyard here. And I say coming back around because this is a deal really to facilitate autologous cell therapy. So when you take cells from one patient, manipulate them in some form or fashion, and then re-inject them back into that same patient. So basically making a new drug custom to every patient.
And so this is called, their technology is the cell shuttle high throughput manufacturing platform. Essentially take the apheresis, they load it into the machine, and once you get it out, it's infusion ready. They can do up to 3,000 cell therapies a year with this, and it's unique because it's very adaptable. They can do, the claim was up to 90% of suspension cell therapies with this, whereas a lot of times, a lot of the other manufacturing processes were engineered as supporting this specific therapy. And so if a change were made, you had to go back and you had to validate the platform, right? And so here, there's some engineering and programming features that allow it to be highly flexible so that you can continue to use it for different ones. It's also consumable cartridges there, which certainly helps in allowing the increased throughput and workflow.
Yeah. So very interesting from a capabilities perspective, and it does bring up the possibility of basically the cost of autologous cell therapies coming down, while at the same time other allogeneic therapies are being developed and could be run in a more sort of batch-wise fashion.
Right. We talked about that before, the difference between the autologous and the allogeneic, where, the autologous ones are being taken straight from the patient that they're going to go back into. So the cells are taken out and modified and then reinserted, whereas the allogeneic are cells that are not taken from the patient specifically that is being treated, but are cells that are then modified and can be expanded and used more in an off-the-shelf format.
That's right. And, really, I see this as a good thing and a win for everyone, really, because if we can keep both options on the shelf, then that just sort of expands the tools that drug developers can use to think about what's really going to be best for a given indication or given disease state.
For sure.
Yeah, so definitely optimistic about that. Moving on to investment news. A trend that we've mentioned in a couple of the previous podcasts has been the migration from some oncology indications over to autoimmune indications. And the story we picked out is one of the most recent examples of this, where Cullinan Oncology, a large Boston, I guess you can still call it a startup, but definitely a biotech, is changing its name from Cullinan Oncology to Cullinan Therapeutics. And it is, as it does that, also getting an infusion of additional cash to help it target these new indications.
Yeah, looking at specifically SLE, so systemic lupus erythematosus. And so they've got their CLN 978, which is a bispecific T cell engager. And that they were originally looking at for non-Hodgkin lymphoma. But now, essentially, you can target CD19 and deplete B cells here. And so it's shown great efficacy so far, specifically in a trial. Patients, I did not record the number, but essentially all the patients in the trial had discontinued immunosuppressive treatments, and therapy had been followed through 15-month follow-up. So certainly looking very positive there.
And what I love about this move and this industry shift overall is just that we're looking at diseases based on sort of the biological cause of the disease, mm-hmm. and then being able to think about what medications can impact that cause, and then being able to translate efficiently. So a lot of these cancer medications that are targeting the immune system or tweaking how it functions can mm-hmm. then definitely be used for autoimmune, where it's a similar or another type of immune dysregulation.
Mm-hmm. Yeah, the multi-purposing the tools, right? You've got these tools in the toolbox. What else can they do? And your next item as well. I don't want to scoop you here, but again, goes into moving the tools into a different arena. So I'll let you kick that off.
Yeah, absolutely. So another company in our backyard, so Century Therapeutics, another Philadelphia company, basically acquiring Clade Therapeutics and also moving into autoimmune disease. So another oncology company moving into the autoimmune direction. And likewise, this movement was accompanied by an additional private placement there.
Yep. And so the primary therapeutic that they're looking at here is CLDE 308. And so it's targeting CD19 again, going into autoimmune. The indication here, actually, that one's for autoimmune against CD19. The other therapeutic is CLDE 361. This one, what I was going to mention is the target is myasthenia gravis. And then they have another one for solid tumor. So really extending the reach that Century has now as far as therapies, and adding on to their platform currently. I know Century released information as to their INK platform, and so this is actually alpha beta T cell platform.
Yeah, so it covers both cell therapies and oncology to autoimmune shifts. So definitely an interesting one there. And, as we said with the Cullinan shift, basically if we've got these tools to impact the immune system regulation, then why not apply them to anything that has the same sort of root mechanism for the indication? So exciting to see.
It's really exciting. And, what's really fun is then being able to talk about this with colleagues, right? Getting out there to conferences. And so I feel like you've got something to talk about there.
Of course. So definitely May is conference season. And it's actually, overlapping conference season, and KCAS will be at quite a few of these coming up. So depending on when this podcast fully airs, we'll either be still preparing to go or have just come back from, or some version of the two for a bunch of conferences. So on the list, we've got AAPS WRIB, we've got Cyto, we've got AAPS NBC, and then we're actually setting up our own vendor day in early June, and a couple others are on the near horizon as well.
Yeah, and our guest today, David, had attended WRIB last year. David, your thoughts on the conference?
I think it's a great conference. It's nice that sponsors, vendors, and other CROs all come together in one place, get together and talk about similar challenges in the industry and try to gain consensus around standardization and and validations, especially. Also interesting, I think in the past maybe flow was not included as much just because of the subjectivity into it, and so they're having dedicated sessions now just for flow and potentially even having more focused groups in the general CRO council at the end of the week. So a great conference, and I think it's only improving.
Yeah, I mean, as David says, a place where sponsors and CROs can get together, really talk about the science. One of the CEOs that I really admire is Ed Bastian from Delta. And he has an interesting philosophy on this, that basically there's some things that you can compete on with other companies and some things you don't compete on. So, being the CEO of Delta, he views everything from a 30,000-foot view, literally, and is looking at planes.
That was good. I like that.
Right? His philosophy is you do not compete on safety. Right. So everyone needs to drive safety forward. Anything that anybody in the industry can do to make planes safer is good for everyone. And that can never be a point that you advertise on. You can advertise on the service, you can advertise on communication, but never safety. And I like to think about these conferences in the same way, where we need to identify the places where we as an industry need to collaborate on so that our data and results are robust and everybody understands what the data is telling us, and we can help move science and medicine as a field forward. And then really compete on the parts that set us apart, right? The communication piece, our lab space, the quality of our scientists like David that are in the lab doing the experiments there.
Yeah, it's a great point. Want to make sure that when you're supporting a clinical trial, the data is responsible, right? It's something that you're getting data that helps the patients, and that's what we're in this for, right? And I will say that I remember David coming back from WRIB, and you seemed really excited about all the information. It's probably one of the first times I think professionally I was jealous that I hadn't been there myself because of the excitement you had around that.
I think one of the most exciting things was finding out how everyone else was doing it, and to understand that not only were we at least on par, but even exceeding a lot of our competitors.
Mm-hmm. Yeah, that's certainly a great point. And, we had also attended Cyto last year. This was up in Montreal. And Brian, you had recently shared a lot of the workshops that are going on for Cyto this year that you're attending in Scotland. And again, very interested in learning about the things that are being presented, the improvements in the science and standardization and things of that nature, and just really getting together again, both with clients as well as in competitors, vendors, and learning what's out there and how we can continue to improve.
Yeah. Absolutely. No, it should be a great talk. And as I say, always interesting to see who has sort of a rehashed version of the presentation from last year and who has, potentially solved some of the problems they presented. That's always exciting to follow a story through there.
For sure.
All right. And, maybe that then lends itself in to both the paper and our main discussion topic today around receptor occupancy validation. So for this month's paper, we do have the same paper actually that we were presenting on last time, and we had mentioned that we'll be doing sort of an elongated series to delve into some of the details around receptor occupancy validation. So this is a review paper by Liang et al. Out of the MedImmune group, and it was written a few years ago in Cytometry Part B. And it's receptor occupancy assessment by flow cytometry as a pharmacodynamic biomarker in biopharmaceutical development. Now that this is the second time that we're talking about it, I tried to make sure that I pronounced the title entirely correctly. So we'll see if that works.
And we already did share the impact factor, but just in case you're listening to this and didn't listen to the most recent episode, impact factor is 2.65. Just looking at the number of references that a paper and a publication would have. Certainly cytometry part B, this is the clinical arm. There's also a cytometry part A, very influential within the flow cytometry industry.
And with this paper, a decent portion of it is dedicated to basically how to develop and validate these assays. So, just as a quick refresher from our previous validation talks, the development of an assay is really sort of exploring the parameter space and understanding what is of high importance or low importance for controlling the variability of the assay. And then the validation or qualification of the assay is then once you have sort of a lockdown process from your development steps, then characterizing in great detail what the variability is for your finalized process.
Right. You want to know how much you can depend on the data. And specifically when you go through a validation flow cytometry based, you're looking at the various reportables and determining which are the ones and which parameters you're able to count on and what the variance is there, like you said. And so, David, I know you've done plenty of validations, qualifications here, developed assays. Certainly like to hear your thoughts as to, not just RO, obviously, but performing a validation here.
Sure. I mean, and there's so many unique considerations around an RO assay, and you're basically any flow cytometry assay, the precision of that assay is important. But with this one being a semi-quantitative readout, so the actual intensity of the signal rather than population frequencies, I think is kind of one of the first big differentiators when you're starting to think about an RO assay, and especially something that you're going to try and run for over time, several months and potentially years. The development and the optimization of that is incredibly important. And although I usually at least try and give a quick thought on just all the way back to the beginning, thinking about understanding your instrument and the characterization of your instrument. So understanding if you're on a research use instrument or a clinical instrument, but even a clinical instrument, lasers die, and then an RO assay where you're looking at signal intensity, a new laser change to the equipment could potentially have pretty large effects on the output of the data. And so, not to get into specifics yet, I'm sure we'll get to it later, but normalizing those signals over the course of the project or study is incredibly important. But just instrument characterization, so completing IQ, OQ, PQ, operational and performance qualifications will always be essential. And then once you have that basic piece understood, first thing for an RO assay is trying to understand what type of reagents are available to you. And then that kind of dictates which type of assay setup you're going to go with. And we can jump into that in a little bit, probably a few different types of RO assays that are available. Sample type, thinking about sample type can be incredibly important just because the anticoagulant of the sample can potentially modulate the target that you're trying to understand. And again, just the fact that if you're going to run this assay for several months or years, having good controls set up from the beginning and a plan to how you will bridge reagents and controls over that time can be incredibly helpful too.
Yeah, and you mentioned a really important point for RO assays there, David, in terms of bridging and specifically reagents and thinking about as you develop these reagents what to do. So last time we had covered a couple of the different types of RO methods with both free and total and occupied. How do you view, I guess, the development of that, or what are some of the ins and outs of that development step where you're looking at your primary receptor target there?
So I think, it kind of depends on the drug and then the availability of the reagent. So if you can conjugate a fluor to the drug, oftentimes this is one of the best detection reagents that you can use. It allows you to kind of approach it from the free total receptor or the occupied and total receptor angle. I think if it's an antagonistic drug, primarily you're trying to use the drug as a detection reagent. So the patients have undergone treatment, they have receptors bound with the drug. So anything that you bind with additional drug is considered the free receptors. And then with a non-competitive reagent, you come in and stain for total receptors, and then this allows you to calculate the occupied receptors. It seems to be the cleanest from my experience, and if you can get the drug with high specificity, then that usually allows for the best, most clean data.
And that makes sense. And I guess, one of those development steps too is always the screening. And you referenced this a couple times, if you can get, right? So depending on which receptor you're looking at, how many clones there are, then you could be looking at different epitopes of the receptor that you're targeting. And you can spend as much time or as little time as possible there screening different clones and antibodies and potentially even floors to see what you can get all targeted at the same receptor.
Absolutely. And then the pharmacodynamic changes induced by drug binding, right? So trying to understand that side of things is also very important. So once drug binds, does the receptor solubilize into the patient's blood? Does it internalize into the cell? In which case, these kinds of events can skew data, and it is important to understand which way it may be skewed.
Right. And also, too, if you have drug binding, what you may actually do is target cell killing, right? And so all of a sudden, that target population that you're looking at is diminished. And so it can certainly make it difficult to measure RO there.
Absolutely.
But in talking about the reagents, Brian, you brought up a great point too, is the conjugation. And when you're looking at the different types of RO assay that you have to consider in some of the aspects the conjugation. So PE being so much larger than other fluorochromes, if you're talking about something that is non-competitive and you have the drug bound and then you're trying to get this antibody bound, but then it has PE, for example, you might incur steric hindrance there and would alter the output, the data that you're getting. It may not necessarily be as indicative as if you were to bind it to, say, a FITC or something that's much smaller.
Interesting. And with RO assays, I know, Dave, you were mentioning that, trying to minimize the variation between reagents. So I know a couple times we've used different types of beads to help quantify that as well, right? To actually get the specific number of receptors as opposed to just sort of a relative fluorescent intensity level.
Exactly. Yeah. I mean, so over the course of, several years study, a lot will expire, you'll run out of it, and you'll need to change it. And this is not even just reagent specific. The instrument will also change over that time. So normalizing the signal becomes incredibly important to find potentially small changes that are still significant in patient readouts. So the two main beads that most of the community is using at this point in time are MESF beads or ABC beads and antibody binding capacity beads, and it allows you to, exactly what you said, quantify the number of receptors. It normalizes the MFI readout. So if you get a new laser and the signals are brighter for that laser, the patient signals will be equally brighter, and it can be normalized back to the standard curve of the ABC beads. And the same thing with the detection reagent. If you get a new lot and it's twice as bright as it was, that signal will be normalized so that you don't think that your patients at the second half of your trial have twice the number of receptors occupied just because you got a new lot of reagent or a new laser in your instrument.
Yeah, it's certainly a very sensitive assay, and every step that you start increasing or in adding variance makes it more difficult to interpret the data.
Yeah, I mean, I think humans do a pretty good job interpreting frequency populations, and in the clinical setting, that's how it's been done historically. This is much more of a raw data readout, and so normalizing it and then understanding how the drug is performing and what it's doing to the patient's pharmacodynamics is incredibly important.
Yeah. And, within RO validations, we talked a little bit about some of the development steps that are different. I guess the things that will be the same with an RO validation, we are going to run many of the standard H62 document scripts. So the interassay, the intra-assay in many cases,
Mm-hmm.
the inter-analyst, inter-instrument comparison where relevant. Anything else that would be, I guess, the same with a standard validation? And then, more importantly, potentially, what do you consider as a difference that you would want to see in any type of RO assay that we validate?
I think you got the high level ones on what's similar. There's potentially sample stability and process stability. If it's a frozen assay, you may want to do pre-freezer stability or long-term freezer stability, which are also pretty standard scripts that we'll look at, regardless if it's an RO assay or just an immunophenotyping assay. One thing
that brings up though sample type, right? And so with an RO, it's a delicate balance you have with the binding of the therapeutic to the receptor and what that might do. So I don't want to dive into sample type just yet, but certainly, yeah, you're right about those considerations. If you were to look at stability there on frozen versus if we're talking about the unprocessed stability. So how long is it that the sample is good for before we can't use it anymore to interpret accurately the data?
Yeah, and I do think whole blood assays are probably the most common format here for RO, but can be done in other formats. As far as what's different, one thing that I think interesting, depending on what receptor you're looking at, you may want to look at a larger sample size to understand just the variance of that receptor density in a population. And if one person can have 10 times the number of receptors than another person to start, comparing those patients and how they respond to the therapy may be quite different. So just trying to understand the variance of density receptor and on the specific cell types that you're interested in characterizing.
Now, do you see that as being more important based on the sample type that you would be validating versus, or is it just always you prefer to have more at a higher N there?
The higher N allows you, I think it kind of comes down to more as the target that you're looking at. So if it's a target that's very consistent across the population, we all have similar number of CD40 molecules on our cells. Not so important. If that target that you're trying to quantify varies greatly among the population or the sample that you're looking at, then it becomes more important to try and understand that.
Mm-hmm.
And then the question is always, how high of an N do you want, and who are you using for that N, right? Because I know the qualm about psychology studies is always that we understand the psychology of college students extremely well at this point, and the results are very relevant because that's where all the psychology studies take place. You can pay them $10 and they come and do your psychology study for you. So are we now sort of understanding the biology very well of lab personnel, and then if lab personnel are not representative of the wider population, potentially skewing it that way?
Yeah, that is all. I like that analogy. If disease samples are available, we will always encourage trying to at least do some sort of development work on it, and if it can be included in a validation, absolutely. I think understanding how your assay is going to perform on clinical samples is kind of the main goal here.
Yeah, and the difficulty though with disease samples is how quickly can you get it? They're also costly, right? And so during the pandemic, we were talking about lead times to get fresh disease state samples anywhere from 3 to 6 months. Now, timeline has come down a little bit. Now, to your earlier point about frozen versus fresh, if it's frozen, then generally those are things that you can get pretty readily. Many companies will will stock that. Whereas with the fresh, which is generally the preference of companies performing RO assays, just like as you mentioned, the industry is trying to move in other directions to expand sample types, but in the current state, fresh is preferred. And so certainly disease state isn't always the way that validations get done.
Yeah, and I mean this was a big topic last year at WRIB as well, and I'm sure it will be continued into this year. It's not really in the power of, from our position as a CRO to generate clinical protocols for our sponsors. If we can generate longer term partnerships with them and kind of plan a little more longitudinally, they can actually set their studies up to collect research use samples. And if they're able to do that, in a timely fashion ahead of when they actually want the clinical assay validated, those samples can be used in development and then also in a validation, which, from my perspective, that would be the ideal.
I think talking from the client side, though, time is always of the essence, and there's so much pressure around meeting deadlines that by the time we're getting a validation, it's a mixed bag whether or not there is going to be much in the way of time to be able to get that set up, right?
Of course, yeah, that's always the balance.
Yeah. All right, and that makes sense. So in terms of characterizing variability, getting sort of a wider population view, any other primary differences? I know for cell therapies, we always talk about limit of detection and limit of quantification. Anything else, since this is also sort of semi-quantitative for RO assays, that we generally recommend, don't recommend, always make sure to talk about?
So I think definitely LLOD and LLOQ, maybe we get to that in a second. But before that, I think linearity is helpful and can be important. And it just kind of depends on where you want to do that portion of the work. So it can be done in development and optimization if that's the preference. Understanding, at what concentrations of drug treatment do you get full saturation, and what does that look like compared to an untreated sample? Doing some sort of dilution curve with these doses of drug is important so that, you can understand, are you getting full occupancy? Should you expect full occupancy, no occupancy, partial occupancy? And so, maybe 5 to 12 different drug concentrations, and then looking at the signal intensity and plotting those against each other is important, because you will need to include at least some of those conditions for the even standard test scripts in a validation. So linearity needs to be done somewhere. I don't know that I think we're kind of agnostic as if we want to do that during development or during validation. But that one can be extremely useful.
Right, and you bring up a good point. I mean, when it comes to both the development and validation, there are definitely plenty of things where we provide flexibility there, understanding that, in flow cytometry, there are several ways to achieve the goal. And so in your example specifically, when a test script is going to be done, as long as I would say it could have implications as to the amount of work that goes into the validation qualification, right? Mm-hmm. So if you could attempt to try to do things concurrently, you could pull out the linearity, you could do it earlier, but it's just all kind of baked into that mix there.
Exactly. Yeah, it'll need done at some point, and whether or not you want to, put stringent criteria around it and keep it in the validation, or use it more for just a tool to understand your assay and drug concentrations that would be valuable to include in the validation. It can go either way.
Okay. And then do you have, David, any preferences on control material or control material or a control strategy, I guess, for the assay that you always recommend?
Yeah, so I think you definitely want to include some sort of control material. I think stabilized blood specimens are probably the easiest, most cost-effective way. They're incredibly standardized. Therefore your data is incredibly reproducible. The only issue is that sometimes the signals off of those controls don't mimic exactly what fresh biological samples would look like. But that piece is maybe less important than just having it look the same every single time that you run the assay. Any difference in that control sample should be investigated and try to understand if it's an instrument issue, sample issue, process issue, and so that is generally my preference. That especially in an RO assay, the receptor that you're looking to quantify, that signal should be incredibly stable over time. Another thing that can be added if analyst variability is a concern, you can use lyophilized cocktails. That helps to really keep that signal stable over time. Other options, if for some reason stabilized blood is not an option, like a strep control, cell lines that express the target could be potential options, so that they would also be, the signal intensity would be stable over time, or frozen PBMCs. And in either case, if you're changing from lots, or in PBMC case from one donor to a new donor, doing batch to batch normalization and bridging will be important.
And when you're talking about cell lines, for example, that's where we'd be spiking that into healthy donor blood so that we can mimic the disease state. So trying to make sure that we've got significant or sufficient expression of the receptor of interest.
Yeah. And if it's there are some targets, I think, that really are only being generated in like an immunotherapy platform, and therefore they're just novel targets in general. And the only way to get those to express is to use a cell line.
Right.
And
go ahead, Brian.
wanted to just highlight one thing that you said. Like you mentioned using cocktails to control for a specific type of variability, right?
Mm-hmm.
And it's good to think about control material for exactly that purpose. If you use something like a CD-Chex or a PBMC, you are controlling for one specific type of variability. Are we basically putting the right antibodies in the right tube? Is the instrument functioning sort of as we expected it to? So one control definitely can't do all the different things. So I know we frequently will use FMO or FMX tubes for a gating control, sort of in addition to any type of control material to view the performance of the assay over time.
Oh yeah, absolutely. And for an RO assay, the gating controls are very helpful in getting you to the point where you've correctly recognized populations of interest, but then on those populations, you actually read out the entire detector for the receptor that you're trying to quantify. And that also helps remove that subjectivity of, okay, a human decided to put the gate here, so that's what we're calling positive. It removes that level of subjectivity, which can be helpful.
And just making sure for completeness, I know we've talked about healthy donor samples, disease state samples, spiked cell lines, cell or synthetic standards. And then did you mention stem samples as well? And if not, thoughts on that?
Stem samples can be helpful, I think, if it's the only way to generate a positive control. The challenge there is that getting really consistent QC material stemmed exactly the same can be difficult.
Right. So you'd want to make a lot of that up front to be able to test on consistently over and over again.
Absolutely. I guess the last one is the limit of detection, limit of blank, LLOQ. That piece can be used depending on what type of assay you're doing. That piece is included in other flow cytometry assay platforms, but maybe especially important in an RO assay, just because as patients have more drug bound to their receptors, we actually lose the signal in the flow assay. So having very, very low signals and trying to understand where that signal is reliable versus where it fades into the background is always extremely important and helpful.
Mm-hmm.
And I think there's still some, maybe the industry has not quite landed on a standard way to do that, but basically diluting treated samples that have completely occupied receptors into untreated samples allows you to dilute that signal intensity. And at some point, right, and so if you are doing this with MESF beads or ABC beads, which is the recommendation, there is a blank bead that has no receptors. So at some point, when you theoretically should have available receptors, and therefore you should have signal, it equals the blank, and so at that point, that is your limit of quantization. And for your limit of blank, you actually will just look at fully negative samples. So in the case of if your setup is the free and total receptor, something that is totally occupied, so the signal would look totally negative, would be where you measure your limit of detection and limit of blank.
All right. Nice. And I know we wanted to wait until the next episode to talk about, in detail how multi-site and longitudinal trials work. And I know we covered in pretty good depth the receptor occupancy validation considerations, at least. Anything else that we wanted to mention on the validation considerations for RO assays?
I don't think so. I mean, just reliable data over months and years is the goal, and clean data that's not noisy, and those it's a lofty goal that is challenging, and it takes a lot of upfront considerations, and in our industry, a lot of alignment with our sponsor.
Mm-hmm. Another consideration is that RO assays can still be done on bispecifics, for example, just is something that is a bit more complex there. And so considerations have to be for detection reagents on, both arms, specifically understanding of the receptor biology, both the receptors there in question, whether it's necessary to assess RO on both on a bispecific, or if one would be sufficient depending on the mechanism of action there. It certainly just adds a level of complexity than your standard monoclonal antibody. And
that level of complexity is a good point, Adam. In terms of validating these and treating them as semi-quantitative readouts, our advice to sponsors during development is to, in the vast majority of cases, keep the RO assay relatively simple, because, as you mentioned, even bringing in a second receptor, yes, these flow cytometers can do 40 colors. However, if you do all 40 colors and are trying to look at the variation of one very carefully, it can be very difficult to look through all the noise. So general recommendation is to keep the panels as small and as focused as possible for this specific purpose, and then have potentially a second tube or second panel that's used for broader immunophenotyping if and when needed there.
Absolutely.
Nice.
I'm glad that you brought that up. I was totally thinking about that earlier and then got sidetracked on all the other good discussions. When you said that, it was like a light bulb went off in my head. I was like, oh yeah, going to totally mention that.
Yeah, absolutely.
So David, anything to add on that as far as complexity of panels in RO?
No, completely agree with the sentiment. And, if the question is, we want to understand the number of occupied receptors, let that be the main question, and design the assay around it. And the simpler that you can ask that question, the more robust and reproducible and real the data will be.
Yeah. And that may be a good segue into some of our fan feedback from previous episodes. So for this month's episode, we did get some feedback on one of our previous episodes. So Paul Robinson from Purdue University was listening to our Spectral Flow podcast, which, first of all, is a little bit flattering. So I'm very happy about
Very
that.
much so.
Serious.
Right? Yeah. But he let us know that he listened to the podcast, and he's just really impressed by how quickly Spectral Flow has really come about in the field. He did highlight that, of course, that this came from his lab, and he's got patent US 7280204, and it was first published in 2004. And he highlighted that it was really presented at a CITO meeting that year. So always good to see. First of all, that our podcast is making it out there, and that the meetings that we're attending ideally are some of the source and the first places where we get to hear about some of these new developments.
Excellent homework on your part there. Getting the patent number and everything. That was really awesome for Paul to reach out. So thank you. I definitely can understand and appreciate the level of knowledge and background went into that. Is certainly very incredible there. And just having shared our experiences, right, Brian, David, I'm sure I kind of would love to hear your thoughts on to moving into Spectral. I know I had been very excited about it from the first time I heard about it, just the advance that it provides within Flow Cytometry. But David, tell us about when you got into Spectral.
First of all, I can't believe that it's been 20 years since that paper.
Right?
I mean, yeah, so that actually slightly predates my career in Flow. So maybe I'm not as old as I thought I was. But I think when Spectral was starting to make a bigger splash, maybe more like 2016, 2018.
Yeah.
I think people didn't really know if it was going to take. It was like there was this one company, SciTech, out there trying to put this software together that interpreted the signals differently. And I don't think we really knew if it was going to take hold and take off like it did. Since having had the chance to use some of the spectral instruments across different platforms, I think it's been an incredibly exciting development for the industry. I mean, we were already starting to push to a number of floors and a number of markers in these panels that was becoming incredibly hard to interpret, not even on the data analysis side, but how to generate the data in the first place. And so the advances with the spectral platforms have just been incredible. They continually make running samples easier and more user-friendly, and the data analysis on the back end, which is why we're all running these samples in the first place, has become better.
Mm-hmm.
Nice. But yeah, definitely excited to get that feedback from listeners. Looking forward to more feedback, so please do send in any questions or feedback that you've got, and we're happy to address it on our next available episode there. So I guess that moves us into our last section, just around the pun count per episode and weekend plans. So by my count from our last episode, I counted 2 puns.
Mm-hmm.
And I'm hoping that we exceeded it in this one. I recall at least one that was said, so hopefully there were several others that were included.
You have to be honest, though, Brian. You totally put that. You seeded that one. I have a feeling you knew that pun was going to be added way before we started doing this episode.
Like all good dad jokes, just biding their time. So yeah. Yeah, so definitely proud of that, and hopefully we'll keep increasing our overall pun count per episode here. But other than that, in terms of weekend plans, Adam, what do you have going on?
Going striped bass fishing off the Atlantic Highlands on Saturday, so that's always a good time going out with some family, and striped bass are delicious. Always love catching one and bringing it home and cooking it up, and also just spending time with family there. How about you?
Nice. And of course, anytime you go fishing, hopefully you'll give Todd a full recounting of everything that happened.
Isn't he like by proxy? He's automatically at any fishing at any time now.
It is true. Yes. Yeah. So in terms of weekend, I imagine my 4-year-old daughter has some sort of princess event to go to. So I'll look forward to that. She does now have a pink bow and arrow that shoots arrows with little suction cups on them. So her princesses, I guess, have gotten I don't know, more realistic or more real. I don't know. But she now wears dresses and takes the bow and arrow and shoots them in places. So I'm sure it'll be something involving that.
So you're not going to come back injured, are you?
The suction cup is manageable for now. Luckily, we haven't put on the real arrowheads yet.
And, going back to your comment, I know we've got an inside joke there as far as Todd is concerned, but I'm sure our audience probably doesn't get it. One of our team members, beloved team members, head of QA here, I think, what was it? He didn't actually invite himself down, but he definitely seemed very interested when we had somebody visit on site and they mentioned about fishing. What exactly happened there, Brian?
Yeah, so I've told Todd before that I've never seen so many customer audits where people come out laughing and smiling, but somehow he does it. And I think a decent part of this is through fishing. Yeah. So we had a sponsor on site for a full QA audit in preparation for a clinical trial, and somehow the information came up that basically this auditor's favorite activity was fishing, and it led to a solid 15 to 30 minute aside about how much Todd loved fishing, and then they were inviting each other to different fishing expeditions, and I believe Todd may be going to Florida potentially in the next 1 to 2 weeks to then take up this auditor on that request. So,
well, that auditor had a place like on a lake, and it was a very remote area from what I remember. And man, Todd's eyes, I think, bugged out of his head when he heard about that.
Indeed. So now any fishing expedition, we make sure that Todd is aware. That way we don't lose those sorts of opportunities there.
Fair. How about you, David? What are you doing this weekend?
I am actually going to play my first round of golf for the year on Saturday. And then Sunday, I'll likely do my weekend warrior duties, mow the lawn, and do the yard work. But I think it looks like it might be 80 degrees out, so I'm thinking along with first round of golf this year, I'm going to pull the smoker out and make some ribs.
Oh, that sounds so good.
Yeah, looking forward to it.
You gotta tell me, what are you going to pair that with? I mean, beverage-wise.
Beverage-wise, I'm thinking probably some German lagers because I pretty much exclusively drink German beer. Mm-hmm. Maybe a little tequila if it's that hot. Mm-hmm. Yeah, margarita would probably go nicely with a beer.
Yeah, then that definitely sounds good. So when was the last time you played a round of golf?
I actually didn't play last year because I was having elbow injury issues, and they're almost back on the mend. I'm hoping I can get through a full 18 rounds. It's been 2 seasons since I played, actually.
Mm-hmm. Well, I mean, hey, when we hit Top Golf, you were slaughtering the ball even with a busted up elbow. So
that was fun.
I went to the driving range last weekend, and I had to tell my father, who's like my golf guru, about this. It's like a covered on the top, and I hit the ball, and I was doing really well for a while, but then all of a sudden, every once in a while, the ball would just shoot straight up, and it would hit this wooden ceiling and make a sound like a shotgun or something, and so everyone's looking at me. So
your neighbors love that, huh?
Yeah. It's like, all right, I'm gonna go slink away now.
Goodness. All right. Well, excellent episode, guys. David, thank you for joining us. So next time, as we had mentioned, we will be covering receptor occupancy again, but this time in the context of longitudinal multi-site trials, talking about considerations for how to make sure those go as well as possible and that we get the answers to the research questions that are really at the heart of this pharmacodynamic technique here. So looking forward to that next conversation.
Yeah. Thank you, David.
Absolutely. I was happy to join, and it was fun talking with you guys.
Likewise.